Over 30,000 men die every year of prostate cancer. And the traditional approach to attacking this disease is using hormonal therapies or hormonal manipulations.
Many metastatic prostate cancers rely on androgen to grow and invade and spread. And so blocking it has been an incredibly effective treatment option that really forms the core of how we treat prostate cancer. Unfortunately, cancer cells are under constant pressure to evolve. It sometimes takes years, but almost always resistance is inevitable.
Understanding the whys of why did the cancer become resistant is one of the fundamental challenges that our patients face and that oncologists face as well. So often it means doing a tissue biopsy, which in the University of Wisconsin is something that we can do commonly. However, it's very invasive and very expensive. And you can also imagine that across the country that there are many places that are just not able to do those types of biopsies to understand why did the cancer become resistant. We decided to tackle this question from a very different approach by isolating cancer cells from a blood draw. And by doing that, we were able to extract the nucleic acids or the RNA and transcriptional signature. And as a part of that, we identified that the molecular makeup of the cancer had completely changed from what it was originally. We developed a transcriptional signature that is consistent with discriminating this neuroendocrine differentiation versus the traditional prostate adenocarcinoma. This idea that prostate cancer starts as an adenocarcinoma and is still the lethal form of the disease is something that was really dogma in the field for decades. And it was some really insightful research where people were starting to look at tissue biopsies and find that the molecular signatures show that this was not that original adenocarcinoma, and that's what gave rise to this understanding of neuroendocrine prostate cancer.
One of the ways that we tested our assay is through 2 clinical trials. And what we looked for was if we could find very early detection of neuroendocrine markers in those clinical trials that were supposed to not have any neuroendocrine prostate cancer patients in them. And our hypothesis was that if we were able to find just even a small amount of neuroendocrine prostate cancer markers in those non-neuroendocrine patients, that would indicate that the transformation was underway, even if it hadn't completed. And that's exactly what we found in these 2 clinical trials, is that if you were exhibiting this neuroendocrine phenotype early on, then the typical treatments that we use targeting the androgen receptor were unfortunately not going to work very well or for very long for these patients.
Now, a prostate adenocarcinoma can grow as fast as every 100 days. It could double in size. Contrast that with a neuroendocrine prostate cancer where the tumor doubling rate could be as fast as every 10 days, meaning it could lead to symptoms for that patient even within just a few weeks of time. So that ability to identify neuroendocrine cancer at its earliest stage, before it's causing symptoms, really is absolutely critical, because while we have multiple types of hormonal therapies, if someone has a neuroendocrine cancer, if we give them that therapy, fundamentally what we're doing is wasting time. The advantage of using liquid biopsies, or again capturing cancer cells from a blood draw, is that every site of cancer, every metastatic lesion, does shed cancer cells into circulation. So in some ways, we're actually sampling cancer cells from across the entire spectrum, across the entire body. Now, there may be only 10 circulating tumor cells in one tube of blood. And all the other cells that are in that tube of blood are usually millions or even billions of normal white blood cells or red blood cells. What microfluidic technology allows us to do is get almost single cell sensitivity. So if there's only one cancer cell in that tube of blood, we're able to find it and find those transcriptional signatures of neuroendocrine prostate cancer.
Our assay has shown that we can detect the transformation to neuroendocrine prostate cancer long before we would normally be able to. This presents a really unique opportunity to screen patients for clinical trials or for treatment early.
And this is where we really dovetail with some of the more recent advances in terms of targeted chemotherapies. So new classes of treatments such as antibody-drug conjugates or radioligand therapies, where an antibody is bringing a payload, something that can kill cancer cells, more specifically to that type of cancer, while hopefully sparing some of the normal tissues in the body. So that's the other opportunity for not only identifying neuroendocrine prostate cancer, but identifying it at an early stage when we can knock the disease back before it becomes life-threatening.